people on Zoom hear me if I scream a little bit?
Yes.
Okay, great. So it is a pleasure for me today to introduce you to John Firtina. John is a PhD student at the Safari Group research group at ETH Zurich. He's advised by Professor Onur Mutlu, and he obtained his bachelor and master degrees at the computer engineering from Kent University. His current research interests broadly span bioinformatics and computer architecture topics, including accurately and quickly identifying sequence similarities, hardware and software co-design for accelerating bioinformatics applications, and genomics data analysis, correcting also sequencing errors and developing computational tools for gene editing. He's generally interested in developing new algorithms and architecture for bioinformatics applications to enable fast and accurate genome analysis. And today he will talk about this in his talk. So,
great, thanks. Thanks, Julia, for the nice introduction. As Julia said, the title of my talk for today is Enabling Accurate, Fast, and Memory-Efficient Genome Analysis via Efficient and Intelligent Algorithms. So before getting into my talk, let me start with some, let's say, background about myself and about our group. So I'm John. My pronouns are he and him. I'm a PhD student in the Safari Research Group at ETH Zurich. And as Julia said also, my main research interests are mostly related to solving the problems related to computational biology, accelerating genome analysis, and genome editing. So you can see some of these recent works that we published here, and you can get to know us and reach us using our group's website, and you can also contact me using my Gmail address or via my personal website. So in the Safari Research Group, we are around 40 plus researchers, and like we mainly focus on the problems related to computer architecture, hardware software co-design, bioinformatics, security, and memory. And our motto is think big and aim high. So our group is led by Professor Onur Mutlu. He's a professor at ETH Zurich in the Department of Information Technology and Electrical Engineering, but he's also affiliated with the Computer Science Department since 2015. Previously, he was a professor at Carnegie Mellon University, and he got his PhD from the UT Austin, advised by YalePath, and he worked at like many companies such as Google, VMware, Microsoft Research, Intel, and AMD. We're still collaborating with many of these companies very, very closely, and you can actually reach him using his email address and know more about him using his website, of course. So our current research mission is to mainly build fundamentally better architectures on, again, like topics related to computer architecture, hardware, software co-design, bioinformatics, and security. So we basically take the entire computer architecture, let's say hardware design, into account when designing these hardware and software co-design applications, let's say. So we have in our group 4 key directions. So I will be mostly focusing on the fourth direction over here, which is mostly about the genomics, as you may have already guessed. So we also periodically release a newsletter where we discuss the recent works from our group, and I guess the latest newsletter was released in December 2021. So we also make many materials available in YouTube, including the lectures and public talks. Unfortunately, not this one currently being live streamed, but it's going to be premiered probably pretty soon. We also like make many other materials available, such as the source codes, like the PDFs of our papers, slides, etc. And you can find them using these links. So the slides are going to be available later on, I guess via the YouTube link and some other ways. So I'm going to be talking about the accelerating the genome analysis using intelligent software and hardware design today. So let me briefly give you about like the overview of the research and the problems that we're trying to solve. So the main problem that we're essentially solving is the computing is really bottlenecked by data today. So if you look at the important applications such as AI, ML, genomics, so these important workloads are all data intensive. So basically they require rapid and efficient processing of large amount of data, and actual data is increasing rapidly. So in a way that actually we can generate more than, we can generate data more than we can process today. So for example, the high throughput sequencing technologies is providing us a lot of data growing every year. And this is mainly because these sequencing technologies have become really high throughput and really energy efficient. Actually, this is happening in a way that the genome analysis is becoming like much less costly, faster than the transistors are becoming much less costly based on the traditional Moore curve over here. So as a result, actually, we can generate a lot of data today. And this is pretty good because sequencing, being able to sequence like many genomes has a really pivotal role for making like breakthrough, let's say, discoveries in the genome analysis, such as like discovering, so let me see if there are any issues. Okay. Like for example, like discovering like novel diseases, or like better understanding the outbreaks, etc. It is really important to generate a lot of data using the high throughput sequencing technologies. However, we're really bottlenecked by how fast we can process this data today. And this is not because of the sequencing technologies that like where we're generating data. This is because the software and the hardware where we're trying to analyze the data that these sequencing machines are generating. So in our group, we're looking into a lot of approaches where we can co-design hardware and software so that we can reach the throughput of the sequencing technologies so that we can rapidly process the genomic data and analyze them so that we can quickly find answers, especially for the life critical, let's say, scenarios. So today, data movement dominates the performance because the large chunks of data need to be stored in the storage systems once they are produced. And furthermore, that data needs to move between the storage systems, main memory, and the CPU so that we can process them, like we can analyze them. But data movement is basically very costly. It's very costly such that like computation is a lot cheaper than moving the data. So for example, when we look at when we profile the read mappers, so we see that around like 60% of the execution time for read mapping is spent for read alignment, which usually consists of like the alignment part itself and the seed chaining part. So this is of course because sequence alignment is costly because we use DP-based approaches, but there is also one more reason for that. What we also observe is that most of the times actually we're doing a redundant work. So meaning that perhaps like when we're trying to come up with efficient, let's say, algorithms, probably we're optimizing something that shouldn't exist. Meaning that like we're doing so many of these reads are being tried to map, but they are not being mapped, let's say. So this is basically causing a lot of unnecessary data movement and waste of compute cycles. So if you are curious about learning more about like the overhead that we have and the challenges that we have with the current sequencing technologies, such as the nanopore sequencing technologies, you can take a look at this paper. But as I said, the problem is really the analysis of the data rather than the speed that we generate this data. So we need actually the intelligent algorithms that minimize the redundant computation and also the intelligent architecture so that we can process this data efficiently and quickly. Right. So in our group, we're looking at in-memory technologies where we can process the data where the data resides, or near memory technologies such that we can process the data very close to the memory itself. So this enables us to either avoid or minimize the data movement costs so that we can make the computations quickly and efficiently. So you can read this paper more if you want to learn more about the challenges in designing the specialized hardware for genome analysis. So again, like we produce many works in this area, like and for achieving fast and quick genome analysis. One of these works are, for example, like Sneaky Snake is one of these works, where basically we quickly, without performing the read alignment, we quickly try to filter out the reads that are not going to be mapped eventually, so that we can reduce this redundant computation that you saw previously, which was like 98% of the execution, 98% of the read alignment part. So this is this Sneaky Snake is developed for CPUs, FPGAs, and GPUs, so that we can perform some filtering before doing the read alignment. And Genome is another work that we published from our group. So this was presented in Micro 2 years ago. So we developed a hardware software co-design for performing approximate string matching using the BiTAP algorithm. And we show that Genome, which is the work that we published, Genome can actually provide significant speed ups and also the energy improvements compared to state-of-the-art lead mappers. So this is another work that's going to appear in ISCA this year. This is basically Seagram is using the fundamentals of Genome for sequence to graph alignment, and we achieve again significant speed ups and the energy improvements for performing sequence to graph alignment. So GenStore is another work which was presented in US+ this year. So in GenStore, we observed that actually we don't really have to move the data that frequently to from the storage systems to memory, because most of the data can be processed within the SSD itself. So we developed some hardware design for quickly identifying the similarities between the sequences and the reference genome, or dissimilarities, without moving the data, which basically reduces the data movement cost a lot, which eventually improves the performance of the applications that use sequence alignment. So essentially, we believe that the future is going to be more focused on the genomic data analysis, as we basically produce more genomic data quickly and also probably cheaper. So we believe that it's really important to develop architectures that are both energy efficient and also quick, so that we can analyze this data, especially even for like the mobile, let's say, platforms, which are really important for certain applications. We also do the analysis of the existing works. For example, this work is a survey that analyzes let's say all the efforts that's been done since, I guess, 1980s for read alignment. So this is a 111-page work. So if you are curious about learning more about like what are the common methods in read alignment and read mapping, so you can read this paper. This is also another lecture that I can point you to if you want to learn more about again, like the genomic related works that we're doing in our group. Let me see, sorry. Some again lectures. So in general, actually, we believe that the emerging technologies can play a pivotal role for many applications, and one of these applications are, of course, genomics related applications. So if you also want to learn more about these emerging technologies, you can take a look at this paper. This is the first analysis of the first commercially available processing in memory technology. So you can take a look at this. And this is more about the processing in memory and accelerating important applications. So, but like clearly we need both intelligent algorithms and intelligent architectures to handle the data well, let's say, so that we can quickly analyze and come up with the results, especially for genomics applications. So this is all about the overview of the works that we're doing in our group. So I'm going to be today talking about these 3, or you can say 4 works, and they're all mostly on the software side, except for the ApHMM, and I'll start with Blend. So blend is a mechanism for finding approximate seed matches. So it's available in arXiv as a preference, so you can access it. You can access the source code as well. But before that, let me give you some background on seeds. So the goal of using seeds is to quickly identify matching subsequences between genomic sequence pairs. Because we do this because we don't really want to basically align each genomic sequence pair to each other because it's very costly. Rather, we want to quickly identify the matching blocks between DNA sequence pairs so that we can start from there. Basically, it becomes a clue for us. So you can assume that like we have these two sequences, right? And we want to identify whether these two sequences are similar to each other. So how we usually start in read mapping or for seeding applications is that we identify some short subsequences within one of these sequences. And like identifying these short subsequences are known as seeding approaches. So you could be using any seeding approach that you prefer. And yeah, these are called seeds. And what you do is that we use any hash function with a low collision rate in order to generate the hash values of these seeds, and such that we store them. This is one way of it, one of the most common way of it, such that we can store them in a hash table. So this step completely, like the complete step of it is called indexing. And we store this because we want to quickly identify whether like other sequences have these matching blocks within them. And in order to identify them, we again perform this similar operations, like we find the seeds, and then we use the same hash function to generate the hash values. But this time, rather than storing these hash values, we query the hash table with the hash values that we find, and then so that we can quickly identify whether there is a match. For example, this one is matching within the hash table or don't match, so that we can find that there are matching blocks between two sequences. So the seeding approach altogether, the indexing approach, helps us quickly identify the similar matching blocks between sequences so that we can quickly say that, okay, these two sequences are closely similar to each other, and maybe then I'm going to perform read alignment between these sequences. So yeah, as I said, the hash tables enable finding seed matches with a single lookup of hash values. So the main attribute with the hash tables is that we can find exact matching seeds based on their hash values because we use low collision rate hash functions, right? Meaning that almost every seed is going to have a unique hash value, let's say. So meaning that we're going to be finding exact matching seeds. There are basically many seeding approaches. One of them is called minimizers. So this mainly samples k-mers based on their hash values. There are space seeds that ignore characters at certain positions to tolerate some substitutions between seeds, or there are linked k-mers, some of them are known as strummers, which ignore gaps between sampled k-mers so that you can tolerate both indels and substitutions. So what is the problem with that? So all of the seeding approaches use hash functions with low collision rate, right? So as I mentioned, seeds must exactly match to find seed matches using their hash values because of the low collision rate. So this is not like completely a bad thing, right? Because we wouldn't want to have we wouldn't want to have the same hash value for totally different seeds. So this is a good thing in order to identify the dissimilarity. But we also probably have completely different hash values for highly similar seeds, meaning that we're actually missing these highly similar seeds when we're trying to identify the similarity between sequences, right? So this is problematic because today the length and the number of seed matches dramatically impact the performance and the accuracy of the read mappers. So we identify these parameters based on this exact matching attribute, which then has a dramatic effect in the performance and the sensitivity. So existing attempts optimizing seed matches suffer from either increasing the use of costly sequence alignment step because they find too many too many similar sequences, but they end up like not being similar when we perform read alignment, or they suffer from limited sensitivity when finding seed matches. So our goal is to enable finding fuzzy, or we could say approximate matches of seeds with a single lookup of hash values of these seeds. So this is critical. We want to be able to do it with a single lookup. And also another goal is to provide a mechanism for any seeding technique. So we don't really want to be limited by a certain seeding technique to generate the same hash value for highly similar seeds, not only for exact matching seeds. So to this end, we propose Blend, which is a fast, memory efficient, and accurate mechanism that can find fuzzy seed matches. So the key ideas in Blend is to exploit the key characteristics of the SIMHASH technique. So I'm going to mention more about SIMHASH technique later on. But using the SIMHASH technique, we can generate the same hash value for highly similar seeds without imposing exact matches of seeds. So we can also perform fuzzy seed matches with a single lookup while providing some windowing guarantees for minimizers, which is again important, that I'll probably talk more about later on. And another property is that we increase the collision rate for similar seeds while preserving the low collision rate for dissimilar seeds, which is the similar property for local sensitive hashing methods, let's say. And another key idea is that we provide a mechanism for converting seeds into sets of chimeras to use these seeds with the SIMHASH technique. So what is a SIMHASH technique? So the SIMHASH technique is mainly used for detecting near duplicate items, near duplicate sets, let's say, mainly for web crawling by Google. So the insight. So, yeah, sure. So the question was, what does it mean moving from seeds to set of chimeras? So SIMHASH technique, as I'll actually talk more about it, SIMHASH technique requires what it does is that it provides you the functionality to generate the same hash value for two sets using the items of that set. So we need to basically move from the notion of seed, and then we should somehow basically represent the seed as the items of its chimeras.
Like a seed could be two chimeras, so you want to move from one seed to the set of
chimeras. Exactly. So we're breaking the seed down into its items. So the insight is that two sets are similar. We say they are similar if their content is highly similar, right? So the goal with SIMHASH technique is to use this insight to enable generating the same hash value for highly similar sets. So the input for SIMHASH technique is the hash values of a set of items. For example, the set could be a sentence and its items could be words of it. And the output would be the hash value for the entire set. For example, a hash value for sentence. So let's do it with an example. So this is an example sentence, and then we can consider that this sentence is the entire set that we're trying to generate a hash value for it. And the items of that set would be its words. And what we first do is that we generate a hash value for each of the words. So you could be using any hash function for that. And later on, what we do is that we do a bitwise weighted sum for each of the bits at every position. So meaning what we do is that actually we find the net difference at each bit position. We find the net difference between the ones and zeros, meaning that so we count the number of ones at a certain position, and then we count again number of zeros at a certain position, and I take the difference, right? So we do it bitwise, and we assign 1 for each one, and we assign -1 for each zero, so that it becomes a bitwise weighted sum. And the result essentially shows us the difference between the ones and zeros at a certain bit position, right? So in this case, for example, here we have, so at most least significant, let's say third bit, I guess we have 8 ones if we count over here, and 4 zeros, meaning that here the corresponding position should be 1 when we do the bitwise weighted sum. So why is it important? Like, what does it tell us? So when I see that this number is positive, so I can directly tell that there were more ones than zeros at a certain position. And if it is negative, then there were more zeros, right? So we use that information in order to generate a hash value for the set, such that we assign 1 when we see that the sign is plus, and we assign 0, and we see that the sign is either is negative or 0. So meaning when I check at a certain when I check a certain bit position, right? If I see a 1, just checking this hash value, I can say that the bits at that certain position, when it is 1, I can say that there were more 1s than 0s. So this is intuitively important in order to generate the same hash values for highly similar seeds. Why? Because when we have 2 sets, then their content is going to be highly similar, meaning that they are not going to generate the same summation result, but they are going to generate the similar summation over here. Meaning, again, probably if it is positive, it's going to stay positive, and if it is negative, it's going to stay negative, except, of course, for the edge corner cases where we're close to 0, right? So this is important, like this is an important attribute of the sim hash technique so that we can generate the same hash value for similar items.
So given sufficiently long sentences, you would expect all of them to be near 0, would you not?
Sufficiently like similar to sets? Is that the question?
Since every bit has a 50-50 chance of being 1 or 0, given a good hash function, given a sufficiently long sentence, you would expect the summation to be close to 0 in every entry, right?
Perhaps, maybe it depends on the hash function that you're using as well. But based on our analysis and the observations, what we see is that actually many of the bit positions are going to be far away from the 0. Although, as you said, like there are going to be some like certain bit positions that are going to be very close to 0. Meaning that if we are, if there are 2 sets such that at these positions where we are close to 0, and these 2 similar sets are generating like the different hash values between 2 sets are actually kind of not agreeing for that position, yes, definitely, like eventually we're going to end up generating a different hash value. But if they are agreeing, let's say, for the position that where we're close to 0, then we can still generate the same hash value for these 2 similar sets, although like they are close to 0. So being, of course, close to 0 is a problem, but not always a problem if like these 2 sets are agreeing at that position, meaning that they have the same bit at that position. Yes. Yeah. So the sentence will be seed, and the words are the k-mers of that seed. That Julia's question. Does that answer the question? Was that Daniel?
Yeah, it answers the question. I find it surprising. I would have assumed that as the length of the sentence approaches infinity, all of the sums would approach 0. But
yeah.
I mean, you know, as long as you're using finite length sentences, that might not be the case. And so in the real world, it could be quite useful.
Yeah, that's a quite good point. So I don't have any intuition with that. This is true. I mean, if the sentence is quite long, in our case, which is not, luckily, that could be the case, definitely. Yeah, I agree with that. Yeah. All right, I'll move on. Julia, by the way, if there are any questions in the chat, could you let me know? Yeah, perfect. Okay. All right. So yeah, I mentioned these. So based on that, we use the sim hash technique in Blend, and what we do is that we find the similarities between the target sequences and the query sequences. And the target sequences are usually the reference genome or the chromosomes of the reference genome, and the query sequences would be your read sequences that you generate from the sequencing technologies. And what we do is that, as you also seen previously, we generate seeds and we generate their hash values using Blend for target sequences, and then we store them in a hash table, which is the indexing step. And then later on, when the query sequences come, we again use them with Blend and then query the hash table. This enables us to generate the same hash value, of course, for highly similar seeds, and also enables us to find approximate matches of seeds with a single lookup, because we can use the hash table directly using their hash values. So let me do a walkthrough of the entire thing quickly. Again, like as also Julia pointed out, in our case, our input set is a seed, and the items of that seed would be the selected substrings, k-mers, let's say, of the seed. So the question is, which k-mers are we going to pick from that seed, right? So our goal is to generate the hash value of the seed using the sim hash technique. For that, like, we should really identify the items of that seed accurately. So so this is an overview of like how we generate the hash values, which is pretty similar to what I've shown previously with the sim hash technique. So you can assume that we somehow picked the items in the seed. So you can assume that these are the items in the seed, and then like we use any hash function to generate their hash values. So we encode them, we represent them in a vector because we use some SIMD operations to perform these addition, and then like similarly we generate the counting results, and then we generate the hash values, right? This will be this one will be the hash value for the seed where we identify its contents, and we use that hash value in order to either store in the hash table or query the hash table. So we have we provide two insights how to select the substrings within a seed. So first approach is called blend I. So meaning like immediate include, like we say that we suggest that we should include all overlapping k-mers of a seed in the set for hashing. This is one approach, and we observe that this is mainly a better approach for when you have like a lot of substitutions within your read. For example, you're using PacBio or ONT reads with high error rate. So this is a more preferable approach for these type of things. And what we do is that, so you first define the length of the seed. Let's say it is 20, and then later on you define the length of the substrings that we're going to have within that seed. For example, it's 16. So in these examples, actually, this will be 5, and the substrings will be 3. Now what we do is that we pick for each of the seed, let's say, or the candidate seed, we pick every overlapping k-mer first. For example, the GAC, ACC, and CCG. For this one, these are the all overlapping k-mers, right? And then we do this for every potential seed. I'm saying potential seed because we still don't know yet if it is a seed or not based on the seeding approach, right? So we generate a hash value for that seed sequence. And then we later on, for example, if you're using a minimizer approach, we give that hash value for that minimizer, and then it has the freedom to say that, okay, this is the minimizer seed or not. So this is one approach. Use the all overlapping k-mers within the seed length that you prefer. So another approach is called blend S. We named it because we like use the Strommer approach, the linked k-mers with the gaps between them. So what blend S does is it first finds minimizers using a regular hash function. So minimizers would be, let's say you have a consecutive hash value, set of hash values, and then you pick the minimum hash value, and then that will be your minimizer. Right? So there are some theoretical guarantees behind using the minimizers, etc. So we find the minimizers using regular hash functions, and then what we do is that we link every consecutive minimizer to generate a seed. So this would be our seed sequence. And what we do is that, like, instead of including all overlapping k-mer within that seed, right? So for example, this is one example of seed. So if you merge these 3 next to each other, so this would be the entire seed, and the overlapping, again, the k-mers would be, again, like every, for example, 3 overlapping k-mer. But instead of doing that, what we do is that we take the entire linked k-mer, these ones, not necessarily overlapping, and then we include them within the as the items of that seed. So this basically constitutes the items of the seed that we pick with the blend S approach. And so whatever the approach that you pick, it can be either blend S or blend I. So we identify the k-mers, and then we include them in the set of k-mers, right, for the seed. And then later, again, what we do is like we use this, with the identified k-mer, and then we generate the hash value for that seed. So Blend can generate the same hash value for highly similar seeds, right? As you are probably convinced so far, and we can actually find these fuzzy seed matches with a single lookup of their hash values, and then we can use hash tables for that, so that we can return both exact matching seeds, because we're anyways going to generate the same hash value for exact matching ones, and also the fuzzy seed matches. So let me briefly talk about the results that we have in the paper. So this is our evaluation methodology. So we integrate the Blend mechanism into Minimap2, because why not? Minimap2 is probably the best. Yeah, that's the really engineering masterpiece. So what we do is that we change the sketching mechanism in Minimap2, and then we change it with Blend. So we use both real and simulated data sets. The hi-fi data sets are real, and the yeast data sets are PacBio, and the ONT yeast data sets are simulated, and the Illumina data set is real again. So we have two use cases to show the benefits of Blend. One is read overlapping, and another one is read mapping. So these are the results for read overlapping to show the computational resources. So the figure on above is it shows the CPU time results, and the one on below shows the peak memory results, and we compare Blend with Minimap2 and MinHash approach, and we show the speedups over here. So, and these are the data sets over there. So what we show is that we provide significant improvements in terms of both performance and memory, on average compared to Minimap2 up to 20x, and compared to MinHash up to almost 1400x. So we can also reduce the memory footprint by 3.6x compared to Minimap2, and also 130x compared to MinHash. So I'll talk about, again, more insights about why we're seeing these results, but these are the brief, let's say, these are the observations that we have. And these are the results about overlap statistics. So the one above shows you the average overlap length that each tool is finding, and the one below shows the average seeds that we use to find these overlaps. And what we show is that Blend can find overlaps longer than Minimap2 and MinHash, which is important in order to generate assemblies that are contiguous and are fully accurate. And Blend uses actually significantly fewer seeds to find these longer overlaps, thanks to approximate seed matching approach. So we show these significant improvements in terms of, let's say, the overlap quality, performance, and the memory results, because now we have the approximate matching capability, right? This enables us to play with the parameters, which were strictly, basically, let's say, limited by certain numbers. But since now we can find both exact matching seeds and also some more approximate matches, which provides a higher sensitivity, this enables us to change these parameters in a way that we can see both the performance and memory improvements, and also the accuracy benefits. So again, we perform, we construct de novo assemblies using the overlaps that we find using each tool. And here we show results for each data set. These results on the left show you some insight about the assembly quality, and these show you some insights about the assembly contiguity. And what we see is that we can generate more accurate de novo assemblies in most cases, and the contiguity results are similar to Minimap2. Again, we show the results for read mapping, a similar figure, performance, and the memory usage. And we compare again with Minimap2, LRA, long read aligner, Map2, and SCON LSH. So we see again a speed up slightly lower than what we see from read overlapping, but again we see significant speed ups compared to each tool. But we have like slightly more memory overhead than Minimap2 and LRA. Still close, but we have more memory overhead, and the memory overhead is lower than Minomap and SQLSH. So, let me give you some insights. So, there are also some more results from the paper. For example, we show that the mapping accuracy of Blend is similar to Minimap2, but what we show is Minomap2 generates the most accurate read mapping results. We also use the same parameters because I said, okay, we get the chance to change the parameters, but like what happens if we again use the same parameters with Minimap2, right? So we also do that. And what we see is that Minimap2 provides significant, again, improvements in terms of performance and the memory usage when we do that, but with the cost of accuracy. This is because, again, like it can only find exact matching seeds where we provide higher sensitivity to the table. And another observation is that Blend S is preferred for hi-fi data, and Blend I is more suitable for erroneous reads. Conclusion. So Blend can find fuzzy seed matches with a single lookup to improve the performance, memory efficiency, and accuracy. So I'm saying single lookup because there are also other approaches, local sensitive hashing approaches, for example, min hash. And what it does is that it generates a hash value for the entire sequence, but it's not enough, right? Because what it does is it generates many hash values using many hash functions in order to improve the collision rate. And then it then calculates the Jacquard similarity in order to identify whether these two sequences are approximately close to each other, meaning it cannot find the approximate matches with a single lookup. Another property, nice property with Blend is that we provide windowing guarantees using minimizers, because if you use min hash, again, you're sequencing the entire, you're hashing the entire sequence, right? It's not really providing you the windowing guarantee. So you don't know like where you're going to have your hits of the hash values. But in the case of Blend, so we can have the window and then we can generate the seed from that window. And our assumption is that, so we already know that we can generate the same hash values for highly similar seeds. So whatever the seed that you're using, with the windowing guarantees or without the windowing guarantees, we provide the functionality of approximate seed matching. So efficient, we also show that, observe that efficient fuzzy seed matches provide higher sensitivity and extra room for adjusting the parameters that are previously strictly designed for exact matching seeds. We also show that we provide significant speedups and lower memory footprint, especially when using hi-fi reads. So there is a need for developing special tools, specially designed for hi-fi reads that are recently coming, but still, like we need to rethink like how we're generating those seeds, how we're performing the alignment and read mapping specifically for hi-fi reads going into the future. And another thing is that Blend finds longer overlaps while using fewer seed matches to find these overlaps. Executive summary.
Yeah, just
sure.
Like the hours and 5 minutes. Like, I will be around. I don't like,
Oh, sure. All right. So yeah, the problem is like the exact matching tools suffer from costly seed matches and like limited sensitivity. And our goal was to enable finding fuzzy seed matches with a single lookup. We used team hash technique to enable that, and then we also show the ways how to break the seeds down into its items so that we can use them with the team hash technique. And we show significant speed ups and the memory improvements, peak memory usage improvements, let's say, with the team hash technique. So if you are curious more, you can read the paper and check the source code. So the second item in our agenda today, which is AirLift. So you can stop me, by the way, like whenever you have questions.
Yeah, like I mean, I have a couple of questions, but they're like more general.
Okay, all right.
I just want to, because I think like someone might stay around for like
sure. Sure. Okay. Yeah. Yeah. We have lots of contents for ours, I guess, but we can only fit them. Some of them.
I was wondering if you have any data on how robust Blend's approach is to like single mismatches, or, for example, like what the rate is of bits flipping in the composite seed at a certain identity?
Yeah, that's a very good question, which we also got from the reviewers. So actually what they ask is that, is there a theoretical proof behind like whether we're going to always have the same hash value like between, let's say, seeds, let's say, that have an edit distance of 1, right? Is there like any distance metric that we can use to provide some guarantees? So unfortunately, the answer is no for that, although like I've tried to come up with something theoretical. So it's currently it's all intuitive for now. So what we're saying is that we can definitely generate the hash values for exact matching seeds. So we're not losing anything in that sense. And plus, like we're also providing this functionality. Some of the approximately matching seeds, some of them, we don't know which ones, some of them are going to have the same hash value if they are highly similar. So the good thing is that it's really unlucky, or it's really unlikely that we're going to have the same hash value for highly dissimilar seeds. That's a good thing. So another thing, I guess, which is closer to your question, is so we provide this example in the supplementary materials, although like I probably won't be able to give you the direct numbers for that, is there are certain parameters that you can play with using blend that will increase your chance for having the same hash value for like highly close, highly similar seeds, which like you may end up generating too many similar seeds. Like you may end up generating the same hash value for the seeds that you don't want to probably generate the same hash value. But again, like what we observe is that up to 3 or 2 submatches, blend can easily, or like frequently what we observe is that can generate the same hash value, but more than that, it becomes basically less frequent. But this is all observation. This is all observation from the data and from the results. And this is true for erroneous reads, for hi-fi reads. So we're using blend S, meaning we're not really using the overlapping k-mers. That is actually less sensitive to substitutions and more sensitive to index. It's empirical how we pick it. Seed length varies from 17 to 27, and the k-mer length, or let's say the number of k-mers that we use within that seed, varies from 5 to 11. So we have around like 11 items up to within the set.
So by seed length, is that the same as window size?
Seed length, can you define the window size? Is the same window size in the minimizer case?
I don't know. You mentioned windows earlier. Normally, I think of a seed as a k-mer, but you're specifying seed as and k-mers as different. So, so you've got seed, k-mer, and window, which is one more term than I guess I understand.
True. So seed is the subsequence that we store in the hash table, and we like use the k-mer term, like of course like it's very generic term. So we use the k-mer term to refer to the substrings of that seed, where we use these k-mers as the items of that seed in order to generate the hash value. And the window would be how many seeds that you're going to consider within your window. For example, when you're finding the minimizer, so how many seeds you're going to consider so that you can pick the minimizer seed. So window length is related to seed, and the seed length is again, like as I said, somewhere between 17 to 27, somewhere between that. And window length is somewhere around, for hi-fi data, is somewhere around 50 to 200. For read mapping, it's 50, I guess. And for read overlapping, it is 200. meaning like we consider these 200 seeds and then we pick the minimum one within these, between these seeds, let's say, or candidate seeds, because if you don't choose them, they are not seeds in the hash table.
Okay, thanks. I understand now.
Okay. All right. Any question? Sure.
Yeah. So I noticed the lab systems.
Mhm.
Is this
Sure, of course, yeah. There are some approaches that perform, for example, read alignment using GPUs. Some approaches that identifies overlaps using GPUs. So parts of the entire read mapping or the read overlapping part can definitely be implemented in GPUs. For seeding part, perhaps, I mean, it fits well for SIMD, like pretty basic summations between vectors, but probably needs further investigation if we can partition the data well. Yeah. The question was basically about whether we can use GPUs for these approaches. All right, so second word, AirLift. I'll try to be, let's say, more high level for the sake of time.
Yeah, exactly. Like, I think that, like, you can just like keep kind of overview of the
Sure, yeah. So again, like short background, we know that reference genomes are frequently updated. Recently, we have this SHM13 from Telomere Consortium released. Like basically we're releasing many reference genomes frequently. And in order to basically get the most out of from reference genomes, the ideal would be remap your reads to that entire reference genome again. But that may also become unreasonable to remap all of your reads to new reference genome because of the computational costs. And there are many existing works. What they do is that they quickly identify exactly matching blocks between reference genomes, and whether if these blocks are exactly matching, they simply move the positions from the old reference genome to the new reference genome without actually doing any alignment. So there are these tools that exist, and these are the examples of these tools. And what they do is that they generate files called chain files. It's like a dictionary file that maps these blocks between the reference genomes so that you can move them. So the problem with those approaches is that they can move the annotations or positions only when the same region appears in the new reference genome. But so simply moving annotations between the exact matching blocks are not enough for the sake of accuracy, because what we observe is that, for example, going from HG19 to 38, there are novel, like 77% novel genes that are missed in the HG19. So if you were simply moving the positions between constant regions, like the exactly matching regions, then you will be losing these genes that would affect your accuracy. There are basic potentially important regions that are lost here while moving the positions. So meaning that simply moving annotation is not accurate, as it loses a significant amount of information. So if you wanted to reach the baseline accuracy, which would be the mapping all reads again to the new reference genome, we could basically rerun the read mapping to the entire reference genome. But this is really expensive, as we actually discussed earlier, because like there are many sequencing reads and the reference genomes that we generate every day. So our goal is to be able to maintain the baseline accuracy of mapping reads to an updated reference genome while significantly accelerating the process. And we want to basically enable quick and accurate mapping of every sample, every read from the old reference genome to the new reference genome, whether they are mapped to the exact matching blocks or the new blocks, let's say. And we provide a mechanism for that in order to quickly remap the reads. So what we do is that we design our own chain file, let's say our own dictionary, and then we take the read mapping and the old reference genome. And what we do is that we provide the updated read mapping to the new reference genome so that you won't have to fully map all of your reads to the reference genome. So you can assume that like these are the reads that are mapped to the exact matching blocks. These are fine. Like this is every tool can do. And so what about the reads that are not mapping these exact matching blocks between reference genomes? So we also provide a mechanism to map them. So to do that, we quickly label the regions between two reference genomes to decide if we should move them or remap them. And we basically label them such as like, in order to label them, we generate our index file that we call AirLift index. And our labels are constant regions, which would refer to the exact matching blocks, updated regions, the regions that are updated, but still similar to each other between reference genomes, retired regions that do not exist anymore in the old reference genome, and the new regions are the novel regions in the new reference genome. So there are 8 steps to do that. So we start with basically identifying the exactly matching blocks. So this is again is a common approach. Later, what we do is that for all other blocks that are not exactly matching, we extract the read size k-mers from these blocks. And then what we do is that we align them to the new reference genome. And if they are closely aligning, like many of these k-mers are closely aligning to some region in the new reference genome, we say that these are okay, not exactly matching blocks, but similar enough. So these are the updated regions. And all of the remaining regions would be either the retired region or the new region, if you are looking at the old reference genome or the new reference genome. So, but there is one more step that we should consider. So we didn't actually consider whether these new regions are similar in the old reference genome. So what we do is that we, again, generate the k-mers in that old reference genome, and then we align them. Sorry, we generate the k-mers from new reference genome, and we align them to the old reference genome. And if they are aligning some parts, some regions in the old reference genome, we say that, okay, this is also an updated region. So we should treat it as the updated region as well. So whatever, again, remaining is like these are, if you consider this orange part, these blue parts are exactly matching blocks. And all the other parts are the updated regions. And the other parts basically will be retired and new regions. So we treat the reads that are mapped to the old reference genome previously based on these findings. And we do this only one time for every reference pair, right? This is a one-time task. And we generate that dictionary. And whenever a read mapping information comes, we use that dictionary, right? We say that, okay, is it mapping the constant regions? If so, let's just simply move the annotation. If it is mapping the updated region, okay, then either consider mapping only to that updated region, or again, simply move the position if you don't really care about the cigar string. And or basically if it is in the retired region, right, just remap it fully because we can't decide whether it is similar enough in other regions. And if you have like any other leftover reads that didn't map to the old reference genome, we also map them. So essentially, when you give the read mapping to old reference genome, we say that, okay, there are some regions that you don't really have to do read alignment. These are these regions, so that we can minimize the operations that we're doing for read mapping when we're moving to the new reference genome. So these are the key results: execution time and memory usage. As you may expect, AirLift, our tool, is much faster than the full mapping, of course, and also faster than other related works, cross-map and liftover. These are simply moving the annotations, the positions between reference genomes. Sorry, so let me restate that again. So liftover and cross-map actually performs better than AirLift because like they are doing less computation than the AirLift is doing. But AirLift is faster than full mapping, and this actually comes with the benefits of the improved sensitivity. So neither liftover and cross-map can generate the suitable band files for calling the variants. So AirLift can do. and we compare our variants that are called using AirLift. And when we do full mapping, so we compare them, and what to show is that the sensitivity results are very close to each other. So even if you're doing full mapping or you're doing AirLift, so the accuracy results that you're going to get are going to be pretty similar to each other, although you're gaining a lot with the performance. So this is the executive summary again of AirLift. Right? Remapping is costly. Existing tools miss out of regions. We want to maintain the baseline accuracy. What we do is that we categorize and label each region, each important region between the reference genome pairs, and we decide to either remap them or move them simply based on the labels we identify. And we provide, AirLift provides speed ups from 2x to 6x compared to full mapping reads, and similar accuracy when calling the variants. So you can, again, read the preprint of it. And we have a similar work as well. I'm not going to talk about it, which is actually the C++ implementation of cross-map, which is related work. So we showed that actually moving from Python to C++ makes sense. So it provides significant speed up. So use C++ or C. So last,
John,
Apollo.
We're kind of, I will have to at least go in a couple minutes, and I'm the one who's recording. We're a little over time. I don't know if how this was communicated. We're beyond the 1 hour mark at this point.
Okay.
So I'm just, you can wrap up in a couple minutes, or the recording won't make sense. It's one of the two.
Sure. Yeah. I'll basically wrap up quickly, talk about quickly show you the results or key ideas of ApHMM and Apollo, and wrap up within 5 minutes. All right. So I actually presented Apollo last year to the patient lab, and also to, I guess, many of you. So this is an error correction mechanism. It's like correcting the errors in the genome assembly. So these are the ways how we find overlaps and find layout and then generate the assembly from these. They are erroneous, and we need to correct them. And there are assembly polishing tools that does this. It takes the genome assembly and the reads aligned to it and then corrects them, and Apollo is one of these. So we use profile hidden Markov models. To do that, we actually use a modified version of it, which provides higher sensitivity. And so we generate profile hidden Markov models for assemblies, and then we provide their probabilities associated with the PHMMs. And then these are basically the steps. We align them and then perform some training in the profile hidden Markov model in order to correct these errors. So we compare it to the other polishing tools, and what we show is that Apollo provides the most accurate results, but it is slower. It's slow because of the training steps that it includes. So for that, we provide an acceleration approach, which is called ApHMM. The print is not available yet, but it's going to be available soon. So we provide an acceleration framework for any PHMM-based application. So that these are used in genomics works like malware detection and pattern matching. And PHMMs are useful in order to reduce the amount of computation that you're doing, rather than basically aligning one time aligning every sequence to each other. We just generate a PHMM graph for group of sequences, and then you could basically align a sequence to the PHMM graph rather than each of the sequence. So it's efficient in that sense. Or we can use PHMMs in order to basically correct errors as done in many works. So PHMM is costly because of some time-dependent dynamic programming calculations, or there are basically many iterative approaches, so they are very costly. And we're doing actually multiple iterations of the Baum-Welch algorithm. So it provides massive parallelism, but there are simple arithmetic approaches that we think that they are not really that good, and there's data dependency. So to solve this, we actually observe that the Baum-Welch algorithm is really the bottleneck for many applications that use profile hidden Markov models. And our goal is actually to accelerate the Baum-Welch algorithm for PHMM applications. So for that, we have some key ideas. For example, we use the modified version of PHMM so that we can resolve some accuracy-related problems. So we exploit some pipelining opportunities. Right? Some data doesn't need to be stored in the Baum-Welch algorithm. We just pipeline them. And we improve the cache reuse. For example, this is an iterative approach, so we could store these values in the cache more intelligently. So we use lookup tables because some multiplications are fixed. So we don't really have to compute it redundantly, and we provide hardware design for that. And like integrated, so we have many cores for acceleration. There are some control mechanisms. So we provide, we show that like the ApHMM improves the speed up, energy improvements as well. We also provide a GPU implementation. Our GPU implementation is better than the state of the art. So we also decide how many cores that we should. We use ApHMM, so you can check these in the preprint that's going to be available soon. And like we show that all of the applications that use PHMMs are accelerated with ApHMM significantly, especially for error correction, because it's the bound matches dominates the entire execution time of error correction. For protein family search, we can accelerate. For multiple sequence alignment, we can accelerate. It also, ApHMM provides better implementation than the other like related FPGA work. So accelerating the bound match algorithm provides substantial performance improvements and energy reductions. We provide a hardware software co-design, which provides the best overall performance. Data movement becomes a bottleneck for some steps that we can actually consider designing them for in-memory or near-memory solutions. So our GPU implementation is the first GPU implementation for the bound match algorithm design for PHMMs. So this is the title, and it's going to be available soon again. As I said, this is the links for Apollo if you're curious. So these are some prior works that we've published in our group related to genomics. And with that, I'll conclude. Thanks.